So far, we've talked about how certain perfectly normal genes, proto-oncogenes, can sometimes become oncogenes and contribute to the development of cancer. There is another group of genes, though, that we need to talk about, and those are genes that are called tumor suppressor genes. Again, perfectly normal genes that code for proteins that can help suppress the development of cancers. Remember, proto-oncogenes code for proteins that tend to stimulate cell division. We said that these proteins fall into 6 groups: growth factors, transcription factors, cell cycle regulators, particularly those that are involved in apoptosis, receptor protein kinases and non-receptor protein kinases, and finally, regulatory GTPases. Let's take a look now at the types of genes that code for very different proteins. Proteins that can help suppress the development of a tumor or the development of a cancer. These genes code for a group of proteins called, you guessed it, tumor suppressor proteins. The normal function of these proteins is to restrain or inhibit the proliferation of cells, the rapid division of cells. Now, when an abnormality occurs in one of these genes, or an abnormality occurs in one of the proteins produced by these genes, essentially what ends up happening is you lose that inhibitory effect, and that makes it easier for cancers to develop. We've actually already talked about a tumor suppressor protein. We've talked about the RB protein. Can you remember what mechanism the RB protein is involved in? There is a gene called RB gene, and RB stands for retinoblastoma. This gene was first discovered and described because it plays a role in the development of a particular type of cancer called a retinoblastoma. The RB gene codes for RB protein. And that's right, if you can remember it, RB protein was involved in our discussion of the late G1 checkpoint in the cell cycle. RB protein helps prevent unregulated expression of the genes that make proteins required for S phase. The late G1 checkpoint, if you recall, is going to determine whether or not the cell is ready to move forward into the S phase. And part of that regulation involves freeing up the genes for expression that are going to create the proteins needed for S phase. There's another tumor suppressor gene that I want us to be familiar with, and that's the tumor suppressor called P53. The P53 gene codes for, you guessed it, the P53 protein. This protein happens to be a transcription factor. It happens to be a protein that is capable of binding to the outside of the double helix of DNA and impacting the expression of genes in that area. Now, the P53 protein is normally involved in sending cells out of the cell cycle, so initiating cell cycle arrest, or G0. The P53 protein is also involved, once the cell is in G0, initiating the apoptosis pathway, initiating the pathway that will lead to the death of that abnormal cell. Just to refresh your memory, this slide shows us the role of RB in the late G1 checkpoint. Remember that we can see the DNA molecule in blue on the left side of the slide, and we can see a transcription factor called E2F. And under normal conditions, E2F is bound with this tumor suppressor protein called RB. Now, when the cell goes into the cell cycle and prepares to divide, there will come a time when we're late in G1 and the cell has to go through this checkpoint. It has to assess whether or not the cell is prepared to move forward into S. The events that should have happened in G1 must have already happened, and we're going to need protein production in order for S to occur. We're going to need the enzymes and other proteins for S to happen normally. This checkpoint involved CDK cyclin. Remember, that's cyclin-dependent kinase. Has to be bound to cyclin in order for it to become activated. The CDK cyclin molecule then phosphorylates this tumor suppressor protein, which causes it to release from E2F. E2F is then free to interact with the DNA molecule and promote the transcription and translation of genes that are making proteins required for S phase. So unless this occurs in a cell, the S phase will not occur. The cell will not move forward into S phase. Now, tumor suppressor proteins are really important, and tumor suppressor proteins help prevent cells that are taking on abnormalities, cells that are becoming initiated, that are entering into promotion, into rapid division. It helps suppress those cells from moving forward into cancers. But like all biological systems, this tumor suppressor system sometimes fails. And when tumor suppressor proteins fail, it becomes easier for cancers to develop. Remember, we said a tumor becomes a cancer when it has the ability to spread, when it has the ability to metastasize. All metastasis is the development of secondary tumors at sites other than the site where the primary tumor developed. That's what metastasis is. It's spread. Now, there are things that a tumor cell, a cancer cell specifically, there are traits that that cell must have if it's going to be capable of metastasis. Remember, we said cells go through initiation, they go through promotion, they go through progression to become cancers. They get lots and lots of mutations. And those mutations are going to give them new traits, traits that we don't see in normal healthy cells. Now, the traits that are required for an abnormally dividing cell to become a cancer cell, to be able to metastasize, are the traits I have listed here. These cells have to develop a resistance to apoptosis. If a tumor suppressor triggers the apoptosis pathway, these cells have to be able to resist the actions of the apoptosis pathway. These cells have to de-differentiate a little bit. They have to lose some of their mature traits, their differentiated traits. Sometimes you'll read this described as returning to their stemness, returning back to when they were a less mature stem cell. They have to develop the trait of invasiveness. They have to develop a trait that normal cells don't have, which allows them to invade healthy tissue. They have to be able to survive in the bloodstream. We mentioned this earlier. They have to be able to survive immune cells acting on them in tissue. And lastly, they have to develop the ability to stimulate a process called angiogenesis. And angiogenesis is just the growth of new blood vessels. Remember, a tumor is a ball of cells and it is rapidly dividing. That tumor, as it gets bigger and bigger and bigger, it's going to need fresh blood. And one of the things that a fully metastatic tumor has to be able to do is stimulate the growth of new blood vessels so that it can get the nutrients it needs to continue on with rapid cell division. So to become a malignancy, to become a cancer, the cell has to go through initiation, promotion, progression. It has to survive all of those mechanisms that would normally destroy it, and then it has to spread. The process of spreading is fairly basic. I've listed the steps here. The very first thing for metastasis to occur is some of the cells that are on the primary tumor have to break away. They have to separate themselves from the primary tumor. They have to shed off of the surface of that tumor. Once that happens, they then have to literally travel through the local healthy tissue. They have to invade the local tissue. Again, that's an unusual trait. They have to get to and enter into a local blood vessel or a lymphatic vessel. That's also a pretty strange thing for a cell to do, to sort of eat its way through the wall of a blood vessel or a lymphatic vessel. Once they get into the bloodstream, it's the blood or the lymph that's going to carry them to their new destination. At some point, they have to get back out of the circulation, though. They have to exit that vessel again. They have to enter into some new tissue and invade through it. And when they land in their destination, they have to establish a new blood supply, as we said. They have to promote angiogenesis. I just want to remind us here that what we call the blood circulation and what we call the lymphatic circulation, these two systems are very tightly connected with each other. Of course, the blood-related circulatory system of arteries and veins and arterioles, those are simply smaller vessels, and venules, those are smaller vessels. And then capillaries, that's what you're looking at here, this mesh. Those are carrying blood, but there are similar vessels that are carrying lymph, that are carrying immune cells in a circulating fluid. Those lymph vessels are traveling very close to the regular circulatory vessels. And when you get into these capillary beds, where you have arterial and venous capillaries, they are just tangled in with small lymphatic vessels. So these two systems are tightly connected to each other. On this slide, we're looking at a diagram that walks us through those steps of metastasis. Remember, it all begins with a single cell that was mutated or epi-mutated through the process of initiation. And then it was exposed to something that promoted cell division. And then those cells continued to become more and more abnormal. They were progressing in abnormalities until we had this. We had a primary tumor. We had a ball of abnormally dividing cells somewhere in the body. Now, the first thing that has to happen is some of these cells have to break away from this tumor. That's actually quite abnormal. That's an abnormal trait. Normally, we expect cells in tissue inside the body to stay connected to each other. So having these cells sort of shed off the surface is abnormal in itself. The next thing that has to happen is those cells that shed off have to literally travel through or invade through local tissue. That's also very abnormal. We don't expect a mature cell to be able to move through its environment like that. There are some cells that are capable of moving. We talked about flagella on sperm cells. But we don't expect mature, fully differentiated cells in other parts of the body to literally be able to crawl through normal tissue. At some point, they're going to impact a vessel, a blood vessel, or a lymphatic vessel, and they have to eat through the wall of that vessel to get into the fluid inside. They're then just going to let nature take its course, and they're going to let the fluid and the circulation carry them forward. At some point, those cells are going to lodge. They're going to lodge or get stuck. And at that point, they then have to eat their way back out of the vessel. They have to invade normal tissue again. They have to get established in that normal tissue and continue to divide. And finally, when they reach a certain size in the new location, when this tumor reaches a certain size, they're going to need new blood supply. And those cells are going to have to stimulate the process of angiogenesis. So metastasis is a complex process, a multi-step process. And luckily for you and I, every cell that attempts metastasis is not necessarily successful. There are mechanisms that help stop a cell that has left a primary tumor and helps prevent the development of secondary tumors. Remember, we all have an immune system, and our immune system is present both in the blood, in the lymph, and also in solid tissue. In reality, tumors that are about to become cancers or have become a cancer will release several million cells into the circulation, again, either the blood or the lymph, each and every day. That is just a remarkable number of cells that get shed off of tumors and actually make it into the circulation. Luckily for us, most of those cells will be destroyed, either in the blood or in the lymph. Of the cells that survive their trip through the bloodstream or the lymphatic circulation, of the cells that are capable of surviving what will be an immune assault, we look at about 1 out of 1,000 getting through that. 1 out of 1,000 will be capable of leaving the lymphatic circulation and getting into one of the lymph nodes, or leaving the blood circulation and getting into some other organ. 1 out of 1,000 cells. One of the questions that people ask about metastasis is, why is it that the site where the secondary tumors grow, why does that site differ for different types of cancers? And there's a couple of things that contribute to that. The first thing to remember is that the cancer cell population inside of a tumor is going to be heterogeneous. It's going to be different. Each cell within a tumor is going to be slightly different from all the other cells. Remember, they're gaining mutations with each round of cell division, and they're not all gaining the same mutation. So different cells that shed off of a primary tumor are going to have different survival traits. Some of them are going to make it farther away from the primary tumor than others will. The other thing to remember is that there is an anatomical reason that metastasis occurs in some tissues more commonly than others. Remember, a metastatic tumor can grow anywhere in the body, but it just turns out that most of the time they're growing in a short list of locations. And the reason for that is because of the anatomical nature of the circulatory system. Remember, I said that if a cancer cell can survive in the bloodstream, at some point it's going to lodge, it's going to get stuck, and that will be the signal for it to climb back out of the bloodstream. The place where cancer cells lodge in the circulatory system is in the capillaries. This can also happen in the lymphatic system, in the smallest of the lymphatic branching vessels, similar to the capillaries. A capillary, a blood capillary, is only wide enough to allow one red blood cell to pass through it at a time. It's a very, very narrow vessel. And so when these cancer cells get into capillary beds, they tend to lodge there. They tend to get stuck there. So, depending on where the primary tumor is, it's the first capillary bed that tumor cell reaches after it enters the bloodstream that will be the place it lodges. Now, for much of the body, that first capillary bed is going to be in the lungs. Remember, blood returns from the body to the lungs in order to become reoxygenated. Now, many cancers will first exit in capillary beds in the lungs, but we also see a fair amount of metastasis in the liver. And that's because of something called portal circulation. The blood that picks up all of the nutrients from the gastrointestinal system goes first through the liver, through what's called portal circulation. And there are an abundance of capillary beds in the liver. So stomach tumors, intestinal tumors, colon tumors, colorectal tumors, when they shed cells and they enter the bloodstream, they're going to tend to lodge in capillaries in the liver. Now, the other two places that are quite common for metastasis are the brain and certain bones. And again, that has to do with where the primary tumor is located and where the shedded cells are going to first encounter capillary beds. Metastasis, though, is only successful if these cells make it to a new destination and grow into a secondary tumor. And remember, we said the last step of that, as that cell starts to divide again and produce that secondary tumor, the last step is angiogenesis. Any tissue that is trying to grow, is trying to increase in cell number, is going to need blood supply. Tumors are capable of triggering angiogenesis if they have the right set of traits, if they have mutated to the point where they can do something that normal, healthy, mature cells can't do, which is stimulate the growth of new blood vessels. It turns out that when a cancer cell makes it to a new destination and starts to divide and starts to establish a secondary tumor, once that tumor hits a size of about 1 millimeter in diameter, it's going to need new blood vessels. That's not very big, but that's the size beyond which a secondary tumor is going to need angiogenesis to occur if it's going to survive. Angiogenesis is a really fascinating physiological process. Like many processes in the body, it is very highly regulated because it should only be happening at certain life stages. At other stages, it needs to be inhibited because the delivery of blood and blood products is not necessarily going to be a good thing at all times during the life of an organism. So, normally, the things that promote angiogenesis and the things that inhibit angiogenesis are tightly balanced with each other, and one thing or the other will be allowed to dominate at any particular point in the life cycle of the organism. There will be times when the molecules that activate angiogenesis will dominate, and there will be times when the molecules that inhibit angiogenesis will dominate. If you think about a time when tissue is damaged or diseased or destroyed through trauma, that tissue is going to need to be replaced, and that tissue is going to need new blood in order for that to happen. So the molecules that activate angiogenesis need to predominate. Once that injury is repaired, though, we want to go back into the state where angiogenesis is inhibited. On the right-hand side of this slide, there's a picture and a graph that shows us a little bit about some work that was done in researching the process of metastasis. And this was an important series of experiments for helping us to understand when and why tumors need new blood. This work involved implantation of individual or small groups of tumor cells into the eye of laboratory animals. I believe they used rabbits for this experiment, if I'm not mistaken. So what you're looking at the top here is a drawing of the eye in cross-section. So this is the cornea right here. This is the lens of the eye right here. This ring here, and it's been cut in half, but this ring is the iris that surrounds the pupil. And then over here you can see various muscle fibers that are involved in pulling on or releasing the lens to focus the eye on certain objects. Now, what the researchers did was they took some tumor cells and they put them into the fluid here in what's called the anterior chamber of the eye. They did that in some animals, and then in other animals they took these cells and they placed them down on top of the iris. And then they let time pass by and they came back to see what had happened. In some of these animals, the tumor cells were either gone or they were still floating around inside the anterior chamber in this fluid. But in other animals, those cells had grown into a new tumor. And that new tumor had vasculature coming to it, had new blood vessels coming to it. The difference between the two groups was the presence or absence of tissue where new blood vessels could be stimulated to grow. The truth is, you have to have some healthy tissue around the tumor where blood vessels already exist. And during angiogenesis, those blood vessels will sprout new vessels that will be drawn towards that tumor. That can't happen for a cell that is simply suspended in fluid. In other words, there's no way to call new sprouting blood vessels when there's no tissue around. The graph down at the bottom here was made from these experiments, from these data. You can see days on the x-axis. This is something that takes time. And on the y-axis, you can see the volume of the tumor or the size of the tumor. The group that had tumor cells implanted in the anterior chamber is represented by the purple line. And the group that had tumor cells implanted on the iris is represented by the red line. You can see that through the course of 10 days to 2 weeks, animals that had tumor cells placed in the anterior chamber saw no growth, no development of a secondary tumor. Animals that had cancer cells placed on top of the iris had an exponential growth of tumors. And that involved this recruitment of new blood vessels from nearby healthy tissue. So through most of the life of an organism like a human being, angiogenesis is inhibited. The growth of new blood vessels is not required. Blood vessels are in place in anatomically appropriate locations, and they provide enough blood, enough nutrients, and enough waste disposal for the tissue to thrive. But there are times when angiogenesis naturally occurs. Angiogenesis inhibition predominates when there is no injury or trauma or disease going on. Many proteins are involved in this balancing act, as you might guess. Many proteins are involved in keeping angiogenesis inhibited in healthy tissue. These are proteins that have names like angiostatin or endostatin. There's a whole list of them, and you can find tables listing these angiogenesis inhibitors in textbooks of cell biology. What a cancer cell must be able to do in order to proliferate into a secondary tumor and stimulate angiogenesis is basically manipulate the regulatory system. It has to start to cause a decrease in the expression of genes that produce the inhibitor chemicals, and it has to increase the expression of genes that produce the activator chemicals. Both things have to happen in order for angiogenesis to occur. So let's imagine that cancer cells have arrived and have begun to proliferate in this secondary location, and we have a change in the angiogenesis balancing act. We have a reduction in the inhibitory chemicals and an increase in the stimulatory chemicals. Angiogenesis activation has begun. The kinds of things that stimulate angiogenesis are normal proteins that are called growth factors. Two of the ones that we know are involved are one called vascular endothelial growth factor, which goes by the abbreviated name VEGF, and something called fibroblast growth factor, or FGF. These growth factors have to be produced. They have to come from increased expression of the genes that code for them. And it's the cancer cells that are triggering that. Growth factors bind to receptors on already existing blood vessels, on the endothelial cells that make up these blood vessels. And when the growth factors bind as first messengers, they activate a signaling cascade inside the cell. These endothelial cells essentially become stimulated to divide and to make new sprouts, new blood vessels that will head towards that tumor and provide fresh blood for it. The endothelial cells have to do something interesting as they sprout new blood vessels. Remember, we said one of the strange things that a metastatic cancer cell has to be able to do is crawl through normal tissue. If you think about it, sprouting blood vessels also have to be able to crawl through normal tissue. The way that things do that, the way that cancer cells do that, the way that endothelial cells do that, is by secreting a group of compounds called MMPs. They are protein degrading substances called matrix metalloproteinases. These are enzymes, and they're capable of degrading the material that exists in between cells in tissue, the material that we call extracellular matrix. They're breaking down that matrix, and it allows for these cells to migrate, to move through tissue towards some destination. Remember, the immune system is going to destroy a lot of these cells. The immune system exists not only in the blood and in the lymph, but also in normal, healthy tissue. How do we know about the role of the immune system in preventing most cancers? Well, we know that people who are immune suppressed, and I mean severely immune suppressed, we know that those people develop more cancers overall. We know that people who suffer with HIV and AIDS will develop more cancers, for example, than normal healthy people. The immune system is basically composed of two different and interacting systems. What we call innate immunity or cell-mediated immunity, and what we call adaptive immunity or humoral immunity. There are cells called T lymphocytes, and the T comes from the word thymus. T lymphocytes are a type of immune cell that is essentially on the front line of the immune system, cell-mediated immunity. They are able to identify foreign cells and kill them. And they're looking for molecules on the surface of cancer cells that identify them as being abnormal. Remember, one of the features that cancer cells sometimes develop is a cloak of molecules around them that make them look perfectly normal. Surviving cancer cells, the cancer cells that are able to get away from these T lymphocytes and survive, can also release molecules that simply interfere with T lymphocytes and prevent them from doing their normal job. The adaptive side of the immune system, and that's the side that helps produce antibodies, the adaptive side is also surveying these cancer cells. It's looking for certain molecules on the surface of these cells that identify them as foreign. One of the tricks that cancer cells are able to do, one of the traits that they develop, is the ability to remove antigens from their surface. They simply remove the antigens that would normally be present on the surface of the membrane. We're going to switch gears here and finish up our discussion by talking about a few of the features of cancer treatment. Now that we've talked through the mechanisms that produce a cancer, that initiate and promote and progress a cell into becoming a cancer and spreading or metastasizing to distant locations, we'll focus on just a little bit about how cancers are treated. The treatment options for cancers have historically included surgery for removal of the cancer, chemotherapy to kill cancer cells, and radiation to kill cancer cells. Cancer drugs, or what we refer to as chemotherapy agents, have been used to target different components of cancer physiology. including the fact that cancer cells tend to divide very rapidly. The newest chemotherapeutic drugs are designed to target very specific features of individual types of cancer and to produce less side effects. I'm sure you're aware of the fact that cancer therapy, chemotherapy, has been historically associated with terrible side effects, including vomiting and diarrhea, hair loss, fatigue, and weight loss. The reason that these historically successful chemotherapy drugs cause such terrible side effects is because they are non-specific. They simply target, for example, rapidly dividing cells. So they kill cancer cells, but they also kill normal cells that are also rapidly dividing. The newer chemotherapy drugs are designed to limit those non-specific effects. For example, there is a lot of interest in the development of what are called anti-tumor antibodies. So this is a type of immune therapy or immunotherapy where the cancer itself, the tumor itself, is sampled, cells are grown in the laboratory, and exposed to immune cells that will produce antibodies against that particular cancer. There's another type of new therapy called SERMs therapy. SERMs stands for selective estrogen receptor modulators. Remember, we said estrogen is one of the natural hormones that acts as a tumor promoter. It acts to stimulate cancer cells to divide. What these drugs do is prevent normally circulating estrogen from binding to its receptor and having that effect. It therefore reduces the promotion of initiated cancer cells. Now, of course, SERMs therapy only works on tumors that are sensitive to estrogen. With regards to radiation therapy, there are newer forms of radiation therapy, newer machines that can target radiation more precisely. And there are newly developed drugs that serve as angiogenesis inhibitors and essentially help to starve cancerous tumors in the body. Probably the most exciting area of drug development in the field of cancer therapy is the drive to develop drugs that target very specific mutations in different types of cancer. For example, there is a drug called Herceptin, which targets a very particular mutation called HER2 in certain types of cancer. There's a type of cancer drug called Gleevec, which targets a very particular mutation, a very particular chromosome translocation in certain forms of cancer. This is probably the most exciting area because these drugs have proven not only effective, but have very limited, if any, side effects. The important thing to know about the treatment of cancer is this. A tumor does not contain many, many copies of one type of cell. These cells are heterogeneous. They have varying traits from cell to cell to cell. Individual cells are going to gain mutations differently than other cells. So there is no one kind of cell in a tumor. Now, what that means is that the first round, if you will, of chemotherapy needs to kill as many cells as possible. In the past, we used to treat the patient with one drug to start with, and that drug would hopefully kill as many cells as possible in the tumor. But it would only be able to kill cells that were susceptible to that one drug. The problem with that idea that you first treat with a single drug is that some of those cancer cells will survive that first round, because they're resistant to that drug. And they're going to go on to gain more mutations and more abnormal traits. They're going to continue to grow and survive and spread. And you're going to end up with essentially a case of artificial selection. By treating with that one drug, you're going to select for cells that are resistant to that one drug. That's why the second round of chemotherapy is often less successful than the first. The first round is the best opportunity to get rid of the cancer. So, in modern cancer therapy, the first round of chemo is generally a combination of drugs. The cancer is going to be sampled through biopsy if possible. The cells are going to be evaluated in the laboratory for their combination of genes and their combination of mutations. And the therapy will be tailored to get rid of as many of those cells as possible. Another exciting, promising area of cancer treatment is just the fact that there's lots and lots of research going on. There are lots of investigational drugs that are in research labs right now. And cancer patients can often enroll in investigational clinical trials and gain access to these experimental drugs. More and more, cancer therapy is going to be targeted to that person, to their individual tumor, and less on a much wider, more generalized basis. I do want to mention one other area of cancer treatment, which is also growing in popularity and is also showing quite a bit of promise, and that's what's called palliative care. Now, that term palliative translates as supportive. And sometimes people assume that palliative care means that we've given up on treating the cancer, but that's simply not true. Palliative care, or this supportive type of care, is everything that's done with the patient outside of the treatment that is specifically designed to kill the cancer cells. So anything that's being done with the patient to help support their immune system. For example, what kind of diet they're being fed, what kind of advice they're being given regarding sleep, efforts that are being made to reduce their stress level. Pain relief is a big one, providing medications that help reduce the amount of pain they're in from the cancer. And of course, plenty of social support. The ability to become part of a support group, for example, of patients that are also suffering with cancer. The ability to talk to psychotherapists about the stresses involved in going through cancer. All of these palliative treatments are designed to improve the body's ability to fight off the cancer by itself. The human body and the human immune system are remarkable in their ability to heal themselves. And palliative care is essentially designed to support that natural healing ability of the human body. And studies are showing that the more we support the patient with palliative care alongside the tumor-targeted therapy, the better the outcome for the patient. We'll finish up today with this slide, which just outlines some of the newest information coming out of cancer research laboratories. We are doing a good job understanding the genetic mechanisms that underlie cancers. We've identified over 200 of these oncogenes and mutant tumor suppressor genes that are involved in the genesis of cancer. We are learning more and more about the individual signaling pathways that are going on inside of cells as they are initiated and promoted and as they progress. Scientists are sharing what they're learning in these huge publicly available databases, including one called the Cancer Atlas. Different research laboratories are publishing what they learn about the genetics underlying cancer, and they're sharing what they learn with each other so that different labs don't have to reinvent the wheel. As this and other databases grow, we're learning that upwards of about 60% of cancers are traced back to some kind of an abnormal exposure, some kind of an exposure to a carcinogen, or some other lifestyle factor that is causing epigenetic changes. And about 40% of cancers are simply the result of errors during DNA synthesis. 40% are arising out of simple errors that are made during DNA synthesis and are not caught during proofreading or not repaired. The truth is, if we could avoid all those things that we know can initiate a cell, if we could avoid all of the radiation exposure and exposure to the sun, and if we could stay away from chemicals, and if we could avoid viral infection and parasitic infection, we could decrease cancer mortality by about 50%. But only 50%, because some is coming from plain old-fashioned biological error. And of course, we can't avoid all these things. We can't avoid all these things living on the planet Earth in the 21st century. So, where is the hope? Well, the hope is that we now understand how important a healthy, functioning immune system is to preventing cancer and to treating cancer. We're now learning how to stimulate a patient's own immune system by tailoring therapies towards their particular cancer. We're learning to remove restraints on the immune system, to sort of set the immune system loose on cancers. We're even doing some re-engineering of immune cells to make them more aggressive in terms of being able to find and eliminate cancer cells. So the hope right now really lies in our understanding of the role of the immune system in the ability of a human to eliminate cancer from the body.